All right, yeah. Today we have Adriana with us. Thank you very much for coming. And you would like to talk about, let's say, set of recent publications and studies that you have conducted as part of your PhD thesis, which provide, let's say, a fresh view on how action control in more naturalistic settings is accomplished. So, in a nutshell, what did you do and find out?
So in a nutshell, my project focuses on sensory-motor integration processes, which means the integration of sensory input, so for example, what we see, with motor output, so what we do. And this process of sensory-motor integration is actually far more complex than one might think. For example, if I reach for this cup here on the table, my brain has to send a signal to all the involved muscles in my arm and hand, but also estimate the distance to the cup and the force with which I have to lift the cup up. So there are many different calculations and processes taking place in just a few milliseconds.
All right. So, I mean, okay, this research topic is not that new. There have been very many studies about that since decades. So, what is it exactly that puts your important work apart from what's already there?
So in my project, we focus on a more naturalistic setting, meaning that as a first step, we developed a new paradigm to be used in our lab. Usually in psychology and neuroscience, we rely on paradigms or tasks that require discrete responses. For example, that would be a button press with the left or right index finger. However, we try to get more close to what the brain is doing in everyday life, meaning that we need some kind of continuous paradigm. That's why we use a continuous pursuit tracking task in our lab to run EEG studies on these sensory motor integration processes.
All right, so it is not that you go out in the wild and examine how we accomplish very complex action control in a real naturalistic setting, but it is still a lab-based environment where you then want to get, let's say, your hands on the basic principles in, let's say, a better controlled experiment still. Yeah. But what is it exactly that you found out?
So we mainly focus on oscillatory activity in the brain, meaning that different groups of neurons, often in widely distributed networks on the whole brain, fire rhythmically together to sort of communicate across the brain. Depending on the frequency of this rhythmic activity, we know, for example, of alpha, beta, and theta band activity being associated with different cognitive and sensory motor functions. And in my first study, we focused on the time course of sensory motor integration processes. And what we saw was that in the onset of pursuit tracking, we see sort of a sensory motor calibration process taking place in the brain in the form of a burst of activity in the theta, alpha, and beta band. We mainly observed this burst of activity in superior parietal areas, which can be found approximately here on the brain. And these areas have been associated with the selection of motor responses, but also with the representation of the so-called sensory motor program. Interestingly, after this initial sensory motor calibration taking place, mainly theta band activity seems to persist in the brain. However, there's a shift of the location of theta band activity we observe, as it was mainly located in superior parietal areas in the beginning during these sensory motor calibration processes, and later shifts to more medial frontal areas in the brain that are commonly associated with signaling some kind of error. So, for example, the distance between my hand and the position I want to reach.
Ah, right. So this complex reaching, let's say, or for that to happen, theta band activity is most important, but it changes its characteristic of what's being processed depending on the time where you are in the process of reaching, let's say, or in your case, this tracking, and depending on the brain region. So different forms of neural information can be coded depending on time and depending on different brain regions in one and the very same, let's say, action control task, which I think is very interesting because this has never been shown thus far, but it's also elucidating some more about the background of theta band activity or neural oscillations in general. However, what I think, okay, you can, let's say, approach this problem from two different perspectives. One is, for example, you can go, okay, what's being defined in this process by the object that is to be reached, and let's say the effector that you are using to do this action or to accomplish this goal-directed action. Yeah. So in principle, is it, let's say, the same in terms of neural processes when you look at it, for example, from the perspective of the object or from the effector, or is it different?
Yeah, that's actually the exact topic of the second study we conducted. And there we tried to see how different sources of information, as you just mentioned, are encoded in the brain. And what we see is that it is not the same. So different sources of information, meaning the effector, so for example, my hand, and the goal position or the action goal, meaning for example, the position I have to reach to grab the cup, are encoded differently in the brain. And we see that there is theta and beta-band activity differentially coding these sources of information.
Right.
Interestingly, this coding of information mainly takes place in temporal regions that are known to be part of the ventral stream of perception that is known to be streaming from the occipital cortex to more temporal areas. And here, theta and beta-band activity differentially code these sources of information, meaning that theta-band activity mainly represents the current position that the effector has, while beta-band activity seems to code for the action goal. And this shows us that these different information sources seem to be coded by different kinds of signatures in the brain.
Oh, that's very interesting, because when you think about what you beforehand said, that this Theta-band activity is also about this error correction stuff and specification of the sensorimotor program. I mean, trivially speaking, it could be that Theta-band activity is referring to all the stuff that are under control by you as an agent, whereas other forms of neural activity are, let's say, more coding for what is to be reached without having a clear-cut control over that. This is very interesting, but I'm wondering, okay, this is basic science, and you could in principle translate it also to clinical neuroscience. What's about that?
Yeah, so we know that individuals with autism spectrum disorder oftentimes experience difficulties with fine motor actions, but also with basic sensorimotor integration processes. And although these symptoms are commonly known, it is not yet understood how they exactly develop or relate to autism spectrum disorders. And this is why we think that understanding the neural underpinnings and how sensory-motor integration processes work in the brain actually could help us to understand pathologies, for example, altered functions in autism spectrum disorder. And this is why we also already run a EEG study on individuals with autism spectrum disorder using our new paradigm in the lab, and we are currently still analyzing the data of that.
All right, so there's still a long way to go. Are there any specific other steps that you could talk about that you will take until you maybe translate these aspects into a more clinical neuroscience setting?
So what we are currently doing is that we are running a study on adolescents with our paradigm to see how brain maturation influences these sensory-motor integration processes in the brain. And what we already see in our first small sample is that the differential coding of information in the ventral stream of perception seems to be only acquired in adults. However, we still have to do some research on that and finish analyzing the data before we can make some final things about that. And we also conduct this study on adolescents with autism spectrum disorder to have a look at developmental aspects as well as pathology simultaneously.
Oh, this is really interesting. But yeah, I mean, many things at the horizon and many pathways to go. Yeah, it was very nice that you were here with us. Thank you very much for being here, and thank you very much for watching us. Goodbye.
Ja.